everybody. And my talk is, I will present you our study on the modulation of MS-like disease by regulatory CD11C, CD11B, GR1, myeloid-derived dendritic cells induced by antigen-specific therapy with synthetic multi-epitope protein. So, as you might know, multiple sclerosis is an inflammatory disease of the CNS which characterized by neurological impairment of variable extent resulting from primary demyelination and axonal damage. Experimental autoimmune encephalomyelitis is an acceptable animal model for MS. Today, there is no cure for MS. Several disease-modifying treatments have been offered, however, their therapeutic effects are still limited. And because myelin is the primary target for autoimmune T cells, the ultimate goal for MS therapy would be to neutralize the anti-myelin pathogenic autoreactive T cells without affective normal immune cells. So, CNS-specific and non-specific proteins have been investigated in the context of MS. Of these, the major myelin components like MBP, that means myelin basic proteins, PLP, MOG, MOP, and OSP have been demonstrated to elicit autoimmune pathogenic T cells, which responds in clinical EAE, and reactive T cells against these proteins were detected in MS patients. And therefore, because of this multiplicity of the potential target myelin antigen in MS, it is really hard to determine which for each patient against which protein the immune cells will react. However, if one will try and analyze the primary target antigen in one patient, with time, as the disease progresses, the target may change or expand to other antigens due to autoimmunity. And this all impose major difficulties for immune-specific therapy of MS. So, as shown in EAE in our and other labs, targeting a single epitope is unlikely to be sufficiently effective for immune-specific therapy of disease that is associated with multiple pathogenic autoreactivities. So, in the view of the potential complexity of the pathogenic autoreactivities in multiple sclerosis, the ideal approach would be to concomitantly target all disease-relevant epitopes from all non-relevant autoantigens. And accordingly, we propose the concept of multi-antigen, multi-epitope-directed immune-specific for therapy for EAE or MS, and this by using a disease-specific engineered protein which composed of selected highly relevant epitopes from the different target autoantigens. So for this, we constructed a DNA fragment that was designed to encode sequentially and in one reading frame only encephalogenic myelin epitopes in murines, epitopes that were recognized by MS T cells, and epitopes which predicted with high affinity to HLA-DR2. We actually used overlapping oligonucleotides and PCR overlap extension and amplification, and the DNA fragment was cloned into bacteria expression vector. And here you can see the Comasi stain of the purified protein, which we designated YMSPC. So there are several non-regulatory mechanisms associated with immunotolerance. Here I will talk about mechanisms of EAE downregulation following administration of YMSPC. We recently showed that tolerogenic administration of this multi-epitope protein can effectively and concomitantly target multiple pathogenic autoreactivities. You can see here that administration of YMSPC could very nicely suppress disease induced by this myelin epitope, and we also, when we administered YMSPC, it also could treat this disease. That means when mice were already developed clinical EAE, you can see that administration of YMSPC almost actually resulted in complete recovery of all mice from the disease manifestation. This disease suppression was associated with downregulation of the pathogenic T cells and with downregulation of the pro-inflammatory cytokine secretion like interferon gamma, IL-2 and IL-17, and elevation of TGF-beta, as you can see here. Moreover, administration of YMSPC was also associated with energizing the pathogenic T cells and with upregulation of regulatory CD4 FOXP3 cells like you see here. We also studied apoptosis that in this case was more associated with peptide administration. So far I show you the effect of adaptive immune response following YMSPC administration, like shift from Th1 to Th2, energizing of the pathogenic T cells, upregulation of Tregs, and now actually I will focus I will show you actually data on the effect of regulatory dendritic cells that are actually mediators between adaptive and immune response. So dendritic cells are professional antigen-presenting cells which play an important role in Th differentiation and thus involve the induction of both autoimmunity and tolerance. There are two main lineages of dendritic cells differentiation. The myeloid pathway, which results in three subsets, that one of them is the myeloid dendritic cells, and the plasmacytoid dendritic cells, which are known as strong inducers of cytotoxic immunity, as they secrete high amounts of interferon 1. And recently, another subset of dendritic cells was reported as they directly kill target cells. In my talk, I will concentrate on the myeloid dendritic cells and their regulatory capability. So this population is characterized by CD11C, CD11B expression. In the literature, it was shown that increase in this population in Peyer's patches in experimental collagen-induced arthritis following feeding with oral antigens, it was shown that this population of dendritic cells could suppress autoimmune diabetes in an in vivo transfer model, and more importantly, this population play a role in tolerance and suppression of EAE. So therefore, we concentrated in investigate this role of myeloid dendritic cells in maintaining of peripheral tolerance induced by our multi-epitope protein. So for this, first we injected SGL mice with PLP for EAE induction and treated them with PBS, YMSPC, or with PLP peptide. And as you can see here, while PBS-treated mice developed severe clinical manifestation, administration of YMSPC resulted in almost complete suppression of disease development that was also associated with downregulation of the pathogenic T cells and with upregulation of CD4 FOXP3 cells. We then tested the CD11C, CD11B in the spleen. As shown here, significant elevation of CD11C, CD11B population was tested upon YMSPC administration. All CD11C, CD11B expressed high levels of GR1. And actually here we report on GR1 marker which expressed on CD11C, CD11B population, suggesting a new population. To our knowledge, and after a really careful looking in the literature, nobody showed expression of GR1 on CD11C, CD11B dendritic cells. We here also show downregulation of the co-stimulatory molecules on the CD11B GR1 dendritic cells following treatment with YMSPC. And this suggests less stimulatory and more regulatory function. So we then analyzed CD11C, CD11B, GR1 intracellular cytokine expression following treatment with YMSPC. You can see here that CD11C, CD11B, GR1 dendritic cells derived from spleen of YMSPC treated mice expressed high levels of Th2 cytokines like IL-4, IL-10, and TGF-beta, and less and low levels of Th1, which indicates on-regulatory function. You can see here, surprisingly, high levels of IL-17 were detected upon YMSPC administration. We believe, we're currently trying to understand this, and we think that it might be associated with regulatory IL-17 expression. In comparison, as you can see here, no effect was detected in CD11C minus, CD11B plus, GR1 plus cells. So I now analyzed the proportion of CD11C, CD11B, GR1 in CNS and their Th1 and Th2 cytokine profile. You can see here that following administration of YMSPC, there is strong upregulation of anti-inflammatory cytokines following YMSPC administration and downregulation of the pro-inflammatory cytokines. And you can see it both in the brain and in the spinal cord. And moreover, administration of YMSPC resulted in significantly upregulation of CD4 FOXP3 regulatory cells in the CNS. And the question is whether this subpopulation of myeloid-dendritic cells which show this kind of regulatory phenotype are indeed functional. So for this, we injected SGL mice with PLP and treated them with YMSPC PLP or PBS. Spleens were removed and stained with CD11C, CD11B, GR1. These cells were sorted and tested for the ability to suppress the proliferation of PLP prime lymph nodes. As shown here, while this population had no effect on the proliferation of PLP primed lymph nodes, you can see here that addition of this population, CD11C, CD11B, GR1, derived from YMSPC treated mice, resulted in almost 50% inhibition of PLP cells proliferation. So in light of this efficacy of suppression in vitro, we analyzed the function of these dendritic cells in suppression of EAE in vivo. So again, for this, we injected SGL with PLP for EAE induction. We isolated the spleen, stained them with CD11C, CD11B, and then sorted them and passively transferred them for EAE-induced mice on day 5. So as you can see here, while no effect was received with injection with PBS or with PBS-derived CD11C, CD11B dendritic cells, administration of YMSPC-derived dendritic cells resulted in almost resulted in a remarkably suppression of disease development, suggesting functionality in vivo. And moreover, this passive transfer of these dendritic cells was associated with upregulation of CD4 FOXP cells in determination of the experiment. And the question now is what the efficacy of these cells in reversal of ongoing EAE. So with this aim, we injected SGL mice with PLP for EAE induction, as shown here, administration of YMSPC resulted in complete recovery of mice from the severe manifestation. And indeed, you can see here that Following administration of YMSPC, we can see upregulation of CD11C, CD11B, GR1 detected in the CNS of YMSPC-treated mice. And again, it was also associated with upregulation of CD4 FOXP3 regulatory cells. So what I showed you is that administration of YMSPC is associated with several regulatory mechanisms, which include cytokine shift, anergy, upregulation of Tregs, and upregulation of regulatory dendritic cells. We are currently trying to investigate the mechanism which associated with Bregs. So to summarize, our results demonstrate that immune tolerance induced by YMSPC is associated by increase of CD11C, CD11B, GR1 myeloid-derived dendritic cells in the CNS and spleen. These myeloid dendritic cells exhibited immunoregulatory characteristics, including increased production of IL-4, IL-10, and TGF-beta, but reduced of IL-12. Furthermore, we show here that this population was also capable of inhibiting the proliferation of PLP-specific T cells in vitro and significantly suppressed EAE upon adaptive transfer. EAE suppression was associated with upregulation of CD4 FOXP3 regulatory cells. And when we take all together, these findings suggest that IV administration of the multi-epitope protein results in maintaining of peripheral tolerance and reduced EAE incidence by increase of intolerogenic CD11C, CD11B, GR1 dendritic cells. I would like to thank the members of my lab, and thank you for listening.
Some questions?